Good morning, everyone. I'm glad to present our work on tuberculosis subunit vaccine and vaccine-induced immune memory. I come from China. We all know that BCG can induce protective immune responses in children. However, the protective of BCG waned among population among people grow old. So the protectivity may decline to the threshold around 10 to 15 years old. Novel TB vaccines there are two strategies. One is to develop a vaccine which can induce the most robust and longer protectivity than BCG. Another strategy is to boost the BCG-primed immune responses when the protective declined, so to restore the immune responses and provide protection in adults. Our lab mainly focuses on subunit vaccine. The subunit vaccine has the advantage to boost the BCG-primed immune responses. Moreover, our experiment found that subunit vaccine can provide a longer immune protectivity than BCG. As we talk about the subunit vaccine, the first thing is to think about antigen selection. Here is the EAMMH model proposed by Professor Ying Zhang at Johns Hopkins University. In this model, the yang represents the replicating bacteria in our body. The yin represents the bacteria in dormant stage. The yin and yang can transfer each other. So, according to this model, in our when we infected with MTB, there was a replicating and dormant bacteria at the same time in our body. So the subunit vaccine should contain antigens both from the replicating bacteria and from the dormant stage bacteria. This is the growth curve of bacteria. Some antigens such as ESAT, MTB10.4, MPT64, antigen 85B, and MTB8.4 highly expressed in replicating bacteria. The HspX and RV2626 highly expressed in dormant stage. So our lab have constructed several fusion proteins such as EAMMH, which contains antigen 85B, MPT64, and MTB8.4. After that, we linked ESAT to EAMMH. We call it EAMMH. So the EAMMH contains the antigens from the replicating bacteria. Then we construct another fusion protein, TB10.4 and HspX. In these fusion proteins, one antigen HspX from dormant stage. Our experiment found that the EAMMH can induce the best protective along these subunit vaccines. When we combine with EAMMH and MH together, it can induce the higher protective than EMM. We construct the EMM and HspX together to construct a new multi-stage subunit vaccine EAMMH. The molecule of it is 69 codons. We also call it LT69. The adjuvant we used is DDA plus polar IC. First, I give the results on immunogenicity and protective effects of EAMMH. The mice were vaccinated with subunit vaccine 3 times. BCG was immunized at 0 week as control. And the immune responses was detected at 10 weeks and 28 weeks. After a long time, about 30 weeks later, the variant strain challenged with mice. Our results show that with the stimulation of antigens, the EMMH immunized mice can produce more interferon gamma than EMM plus MH group and the BCG group. Similarly, there are more interleukin-2 produced in EMMH immunized group. After H37RV challenge, the EMMH group can induce the better protection than BCG and EMM plus MH. So our results show that the EMMH is a good candidate for TB subunit vaccine. So our next question is how to improve the immunity induced by EMMH. We know that after TB subunit vaccine, after TB vaccine immunization, CD4 T cells and CD8 T cells were activated. The CD4 T cells can develop into Th1, Th2, Th17 populations, and the meantime, CD4 T cells may activate the regulator T cells. The regulator T cells can suppress the activation of CD4 and CD8 T cell activation. So we think if we can download the Treg cells, maybe we can improve the protection of the vaccine. It's reported that L28B can download the regular T cells. So we detect the effect of L20B following EMMH vaccination. We construct recommended adenovirus expressing L28B and treated the mice when we immunized with EMMH. We found with the treatment of L28B, the CD25 positive, FOXP3 positive CD4 T cells decreased obviously. So it confirmed that L20B can downregulate the Tregs. Then we challenged the BCG 4 to 5 weeks after the last immunization, and the bacterial load in lung showed that IL-28B treatment can decrease the bacterial load in the lung. This shows that IL-20B can enhance the short-term protective efficacy. However, in the BCG-primed and subunit vaccine booster strategy, with the IL-20B treatment, we found the cell-mediated immune responses decreased. With the bacterial challenge, and there is no obvious change for the IL-20B treatment, and we detect the lung lesions in after challenge. I find the IL-28B seems to increase the immune pathogen lesions. So we think IL-20B downregulates the Tregs, but it impairs the long-term immune memory for the vaccine immunization. So our purpose is to induce enough memory T cells and so as to have a good protectivity for TB vaccine. So another two factors was considered for immune memory. One is antigen dose, another is rapamycin. We immunized mice with 3 doses of EAMMH. One is 2 microgram, another 10 microgram, and 50 microgram. We found that 6 weeks after vaccination, there is no difference for these different doses. However, 24 weeks after vaccination, we found the 2 microgram can induce high immune responses than the 10 and 15 micrograms. More important, after H7RV challenge, the 2 microgram can induce higher protective effects than the high dose of vaccine. So we find the low dose of antigen can generate long-term immune responses. Next, we detect the effect of rapamycin. Rapamycin is a drug used in clinic to suppress the immune responses because it can block the IL-2 activation of T cells. Meanwhile, rapamycin also used to treat cancers because it can inhibit the proliferation of cells. We used 2 strategies for rapamycin treatment. One is to use the rapamycin after 8 days, 7 to 8 days after EAMMH immunization. That means the rapamycin mainly play a role in T cell expansion time. Another treatment is long-term treatment. After the last time treatment, we plus 28 days treatment of rapamycin. This should play a role in the contraction of T cell contraction. We find the long-term rapamycin treatment can induce higher immune responses than without rapamycin treatment. With the challenge of BCG, both long-term and short-term treatment of rapamycin can induce a low bacterial load. So it seems that the rapamycin can increase the immune responses. In our study, we treated immunized with antigen, and the T cells made proliferation. Then the T cells contracted. After a long time, only the central memory T cells can survive. So we find the high dose of antigen can stimulate more effective T cells, but it induces less memory T cells. In addition, downregulated T cells by IL-28 also induced more effective T cells and less memory T cells. Treated with rapamycin in the expansion and contraction period can improve the quality and quantity of memory T cells. Conclusion: subunit vaccine EAMMH induced long-term TCM T cell immune response in mice, and its protective efficacy against MTB was even higher than traditional vaccine BCG. Regulation of regular T cells does not benefit to long-term immune responses. Low-dose EAMMH has benefit to generate long-term immune memory. The EAMMH-induced immune memory could be enhanced by additional treatments of rapamycin. I appraise the endeavor of our team and the collaborators and the funding for the project. Thanks.
Any questions?
And what about the use of interleukin 7, interleukin 15 to improve the memory cells response?
Can you say it again?
You talked about the use interleukin 7 and interleukin 15 to boost your memory cell response.
Oh, that's a good question. That's the work we are planning to do in next step. Yeah, thank you.